Welcome to our discussion of neutralization reactions and titrations. In this video, we'll predict the products of an acid-base reaction, describe the conditions at the equivalence point in a titration, and identify appropriate indicators for titrations. The first thing to understand for this section is that a neutralization reaction is the reaction between an acid and a base that produces a salt and water. But in this case, a salt doesn't necessarily mean the table salt that we're most familiar with, NaCl, sodium chloride. In this case, a salt is actually formed from the cation from the base and the anion from the acid. So let's take a look at an example. Let's look at the reaction between HNO3 and NaOH, a strong acid and a strong base. So we're actually going to treat this reaction like any other double replacement reaction. So we're going to combine our insides and our outsides. And when we do that, we get the products NaNO3 and water. Now, if you notice for this example, NaNO3 is our salt because it is made up of the cation, the sodium ion from our base, and the anion, the NO3, from our acid. So let's take a look at another example. In this one we have hydrochloric acid combining with sodium hydroxide. So again, treat it like a double replacement reaction and combine your outsides with your insides, which is going to give us the products of NaCl, sodium chloride, which is regular table salt, and water. Finally, give this last example a try on your own. Pause the video, work it out, and then hit play to check your answer. So you should have come up with the products KCl and water. KCl being our salt because it is our cation from our base and our anion from our acid. The last thing to note about neutralization reactions is that if they occur between equal amounts of a strong acid and a strong base, then both the acid and base at the conclusion of the reaction are said to be neutralized, and the resulting solution will have a pH of 7. This is because we have reached something called the equivalence point, and the equivalence point occurs when enough acid and base are reacted to completely use up each other, meaning there are no reactants left over, and we have a resulting pH of 7. The equivalence point is especially important when we talk about titrations, so we'll be digging into equivalence point a bit more further on in our discussion. A very common procedure that is often performed with acids and bases is called a titration. A titration is a method used to determine the concentration of an unknown acid or base, and it involves a neutralization reaction. In a titration, we want to determine how much acid or base is required to neutralize the unknown. So if we are trying to find the concentration of an unknown acid, we would add a base with a known concentration. Or vice versa, if we wanted to determine the concentration of a base, we would add an acid with a known concentration. The setup typically involves a burette and a flask, and the known standard is added from the burette to the unknown in the flask until the equivalence point is reached. And as we mentioned previously, the equivalence point is the point at which the two solutions used in a titration are present in chemically equivalent amounts. It's also when the amount of hydronium ions are equal to the amount of hydroxide ions, resulting in a pH of 7. That's why titrations often involve an indicator that will change color when that equivalence point or that pH of 7 is reached, indicating that the concentration of our acid and our base are now equal.
Hi, I'm Jared Hyman, an assistant professor of chemistry at Elon University. Today, we will be discussing the proper titration technique using the complete single burette assembly kit available from Carolina Biological Supply Company. This is the first in a two-part video series on titration. The purpose of a titration is to quantitatively determine the concentration of an unknown solution, commonly called the titrand or analyte, by adding a volume of a chemical with a known concentration called the titrant. To mark the endpoint of a titration, an indicator is added to the analyte. Let's review some of the equipment that's needed for a titration. Carolina's complete burette assembly contains the basic equipment to get you started, including a burette, ring stand, and a burette clamp. A burette is a long, narrow, graduated tube used to add titrant. It has a stopcock to regulate the flow of liquid. Notice that the markings on the burette go from lowest at the top to highest at the bottom. The ring stand and burette clamp are used to mount and secure your burette. Other materials you will also need include a small funnel to help you fill the burette, a 125 milliliter Erlenmeyer flask, which works best for titrations. The shape of the flask allows for more vigorous swirling than a beaker or other glassware and minimizes spill hazards. A volumetric pipette and pipette bulb to transfer a known volume of analyte to the flask. A wash bottle filled with deionized water, a beaker or flask of titrant and of analyte, indicator selected for your reaction, a reading card to help read the meniscus, and a sheet of white paper to help visualize the endpoint. To prepare your burette for the titration, it is good laboratory practice to rinse your burette thoroughly with deionized water, then with a small amount of titrant. After each rinse, open the stopcock to allow the liquid to drain out the bottom. If there is a lot of liquid clinging to the walls of the burette, then thoroughly clean your burette and repeat the rinse process. The accuracy of this technique is dependent on the titrant flowing into the flask and not sticking to the walls of the burette. Mount the burette in the clamp, making sure it is positioned vertically and there is enough room to position your flask underneath the tip. Check to see that the stopcock is in the closed position. Insert the funnel into the top of the burette and fill with titrant almost to the top. Filling exactly to the zero mark is not necessary, but you do need sufficient titrant to complete the reaction. Check the column for air bubbles and gently tap to free them from the side walls. Remove the funnel. Place the Erlenmeyer flask under the tip and open the stopcock to allow a few milliliters of titrant to flow through, releasing any trapped air. Rinse the tip of the burette with water. Empty the liquid into a waste container and thoroughly rinse the flask. It is not necessary to dry the flask. Record your starting volume. It is important to accurately read the volume on the burette. First, note that the liquid forms a concave meniscus because the water pulls itself up the side walls of the glass. You should read the volume from the bottom of the meniscus at eye level. Secondly, it is important to read your volume to the correct number of significant figures. This burette indicates volume to the nearest 0.1 milliliters. With any graduated glassware, record the volume to one additional decimal place beyond the smallest graduation. In this case, record the volume to the nearest hundredth of a milliliter, estimating the final place. Use the volumetric pipette to transfer the analyte to the flask. Add a few drops of indicator to the flask and swirl. Place the flask under the tip of the burette. Record this volume. Placing a piece of white paper under the Erlenmeyer flask may make it easier to detect the color change. Operating the burette requires two hands. One hand turns the stopcock, while the other hand swirls the flask. Practice turning the stopcock a few times to familiarize yourself with how quickly the flow starts and stops. It is common laboratory practice that the first titration is an estimate. Open up the stopcock and allow the titrant to enter the flask quickly. Continuously swirl the flask. As the volume of titrant in the flask increases, the color of the indicator appears, then disappears as you swirl. When the analyte becomes a colored solution, close the stopcock. Record the final volume of the burette. Subtract the initial volume from this final volume to get the estimated volume of titrant needed for this titration. Knowing the volume that is a little past the endpoint, subtract 5 milliliters from that number to get the amount of titrant that can be safely added before a slower addition is required. As the estimate titration shows, a dark-colored solution indicates an excess of titrant has been added. The desired endpoint is a pale, faintly colored analyte. To reduce the risk of passing the endpoint, Slow down the addition of titrant when flashes of color begin to appear in the analyte. Adjust the stopcock to slow the flow of titrant to a dropwise rate. Continue to swirl the flask with one hand and keep the other hand ready to close the stopcock. When you notice the indicator taking longer to fade, close the stopcock. Rinse the tip of the burette with deionized water and swirl the flask. Record the final volume on the burette. If the analyte remains faintly colored, then you have reached the endpoint. If the analyte is still colorless, then repeat these steps. Add a drop, rinse, swirl, and record until a faint color persists. Typically, titrations are performed in triplicate. Use the average volume of the titrant required to reach the endpoint for any calculations. You now know how to properly perform a titration. Be sure to check out the second video in this series. In that video, we will go over the proper procedure for determining the concentration of an unknown acid solution using a base of known concentration. Carolina has everything you need for your chemistry lab. Visit us at carolinachemistry.com to see our complete line of products and kits for chemistry.
During a titration, we can measure and then graph the pH change of the unknown solution in the flask. The resulting graph is going to give us something called a titration curve. And typically what we'll see on a titration curve is very little change in pH until we near the equivalence point, and then we'll have a very steep and dramatic change, which will allow us to see what volume the equivalent point was reached very easily and clearly. For example, this graph shows us the pH change as NaOH was added to HCl. And since HCl is an acid, we would expect our pH to start off very low. And then as NaOH, a base, was added, we would expect our pH to rise. And again, also as expected, it rises slowly until we reach that equivalence point, and then we see a very steep and dramatic change right here, where we've reached our equivalence point volume. So on this graph, it's very easy to see that equivalence point was reached at a volume of 38.6 milliliters of titrant. And remember, when trying to identify the equivalence point, you are looking for the titrant volume at which a pH of 7 was reached. Indicators are very important parts of a titration, because an indicator's endpoint should be the same as the equivalence point of a titration, signaling that our hydronium and hydroxide ion concentrations are now equal. In order to select the correct indicator for a particular titration, we need to know the indicator's transition range, or the pH range through which the indicator changes color. When we have a titration with a strong acid and a strong base, our equivalence point is going to be at a pH of 7. So something like a bromthiamyl blue would be an appropriate indicator because it changes color within that range, within the 6 to 7.6 pH range. However, if we have a weak acid being titrated by a strong base, then our equivalence point is going to be greater than 7. It's going to favor that strong base. So thiamyl blue or phenolphthalein would be a better choice because their transition ranges are higher in the base range. Conversely, if we had a titration with a weak base and a strong acid, our equivalence point would favor the strong acid and be lower on the pH scale, perhaps around a pH of 4, so methyl orange might be a good indicator to choose for this type of titration. And this concludes our discussion of titrations and neutralization reactions. Bring any questions you have with you to class.